So good morning, everyone. My name is Nadia Blostein, and I'm hosting and moderating this session today. And I'm really pleased to present a really talented master's student from my lab named Nadia Blostein, who will be giving a talk on some work that started a few years ago now, but it seems to be ballooning and ballooning because every time we ask one question, we seem to want to ask a bunch of other ones relating cross-species differences, heritability, and behavior kind of in one interesting package. So Nadia is an MSc student in the IPN program at McGill, and she'll be giving the talk today. Thanks a lot, Nadia.
All right. So hi, as Millar said, so I'm a student at Millar Chakravarty's lab. And basically the crux of this project is to examine heritability, cross-species differences, and latent variables of behavior in the shape of the striatum, thalamus, and globus pallidus. And all of this is going to be done in MRI data. So as you can see from these MRI scans going from the human to the chimpanzee to the macaque to the marmoset and down to the mouse, there are very salient differences between all of these brains, such as the prominent prefrontal cortex in the human, or also the fact that the cortex gets less and less convoluted as you go from the human all the way to the lysencephalic mouse. So although we have identified qualitative differences here, it is of interest to try to examine these differences quantitatively. And so the following graph illustrates the phylogenetic, so evolutionary relationships between all of these species that all have common ancestors. And although the mouse is the only species presented that is not a primate, it still does have common ancestry with primates and is very important to mention within the context of comparative neuroanatomy because rodents are so essential to neuroscience research. So given that all these animals are phylogenetically interrelated, cross-species comparisons can be very valuable in trying to understand what sort of environmental and genetic factors cause these evolutionary neuroanatomical divergences. This in turn is valuable because comparative neuroanatomy studies can help translate the findings from animal studies back to humans. Animal studies are so important because first of all, less variability in their neuroanatomy allows for smaller sample sizes, but way more importantly, animal models allow for extreme experimental control, so over the genetics and environments with which you're working. Also, comparative neuroanatomy studies can help inform neurodevelopmental studies. They can improve our understanding of neuroprotective mechanisms that animals have, which humans do not. And they can be a valuable proxy for neuroanatomical evolution, since currently it's impossible to conduct longitudinal and in vivo neuroimaging studies on a time scale sufficiently large to capture evolution. So as you can see from these brains that are all up to scale relative to each other, scaling really does matter in cross-species comparisons, which is why allometry, which is the study of how the magnitude of some part of an organism varies as a function of another part of that organism, has become seminal in comparative neuroanatomy. For instance, here we have a graph where on the y-axis we have brain weight, and on the x-axis we have body weight. And so in red over here we have the human brain to body weight ratio, which is fairly high, and then below the red line we have the brain to body ratio of fish and reptiles, which is fairly low, as one would expect. You don't, however, need to constrain yourself to examining neuroanatomical structures with respect to body weight. You can also examine their size with respect to each other. For instance, here we have a graph where on the y-axis we have neocortical volume, and on the x-axis we have total brain volume. So the dotted line represents a scaling factor of 1, meaning that the neocortical volume increases at the same rate as total brain volume increases across species. And what's interesting to notice here is that for the human, as well as for a lot of non-human primates, neocortical volume does increase slightly more rapidly than total brain volume, which makes sense given the association of the neocortex with higher order cognitive function. So here we have the allometric curves of the volumes of different brain structures relative to total brain volume across species. So every dot on this plot is an animal. And what's interesting to notice here is that for the neocortex, which is once again associated with higher order functions, the neocortical volume increases more rapidly relative to total brain volume than other structures such as the cerebellum in blue and the striatum in pink, which are associated with more lower order functions. And finally, we can also see how relative brain structure size seems to adapt to the environmental needs of an animal as well as its behavior. For instance, the total volume of the 10 largest auditory nuclei in the brain relative to total brain weight is fairly small in the human, but it's fairly large in the bat, cat, and deer, which is very interesting given to the functions that the auditory system has in all of these animals. However, we do live in a very corticocentric neuroimaging world. So there has been a lot of research examining cortical differences across species. For instance, we have this seminal work by Katia Hoyer and Roberto Toro, which examines cerebral volume and several cortical measures in 34 different species. Professor Vock has also examined gradients of the organization of the structural covariance of cortical thickness in macaques compared to humans. And Riordan and his group observed that a significant positive relationship between the relative expansion of cortical area in the human compared to the macaque and the vertex-specific areal scaling relative to total cortical area. But expanding on this further, we can go back to how genetics and environment can explain certain cross-species and evolutionary neuroanatomical divergences in order to compare heritability maps to cross-species difference maps. So heritability is defined as the proportion of variance of a trait, so variability, associated with additive genetic as opposed to environmental effect. More intuitively, it could be thought of as a quantification of nature versus nurture. And one concrete example of heritability would be height. So it's been shown that height is about 79% heritable, which would explain why tall people tend to have fairly tall children. So going back to the cross-species difference maps that Riordan computed, so looking at how the cortex area is expanded relative to in the human relative to the macaque, we can compare it to a cortical heritability, areal heritability map. And, we can qualitatively observe certain trends emerge. For instance, there's increased heritability and less cross-species expansion in visual areas of the cortex that are usually associated with pattern recognition and reaching and grasping movements. On the other hand, we see decreased heritability and more cross-species expansion in the paracentral lobe, which has been shown to be associated with the control of lower extremities. So this is an interesting finding because feet have a very different role in the human versus the macaque. So we can see that associated part of the brain seems to be quite environmentally adapted in both species. However, the brain is not just made up of the cortex. Three pivotal groups that often go understudied are the striatum, thalamus, and globus pallidus. And going back to our original image, it may not be so surprising that they're less popular in comparative neuroanatomy, given how similar they look across species, or at least when you compare them from the human to the macaque, they look very similar. In the marmoset and the mouse, they look quite different, to be honest. But, and they also only represent around 2.45% of total brain volume. However, if you actually think about the fairly well understood basal ganglia loop and its role in integrating information throughout the entire cortex, it may seem unfair for this group of structures to go so unnoticed. So just as a reminder, the striatum, thalamus, and globus pallidus modulate cortical activity through a direct loop as well as an indirect loop. And their functionality can be summarized into 4 canonical loops, so 2 motor loops, the body movement loop, as well as the eye movement ocular motor loop. And then there's also 2 non-motor loops that they're associated with. So one loop associated with the initiation and termination of cognitive processes, and another loop associated with emotion, motivation, and mood transition. So evidently, these structures are pivotal to a plethora of brain function, and abnormalities in these structures are associated with a lot of behavioral abnormalities as well as brain diseases. So just to summarize what I presented so far, neuroanatomy is at the intersection of many factors that also influence each other. So the 3 that we are focusing on today are genes versus environment, evolution and adaptation, as well as behavior. And so given how important yet underrepresented in the literature the striatum, thalamus, and globus pallidus are, we decided to examine the interplay between all these factors in those 3 subcortical structures. So the way we examined genes versus environment was by using a heritability model, which I'll explain in more detail later. And then the way we examined we use basically cross-species differences as a proxy for evolution and adaptation, because once again, we unfortunately cannot collect data on an evolutionary timescale. And then in order to examine behaviors, we extracted latent variables of behavior through partially squares correlation analysis. And so the large question we were asking through this work was, what is the relationship between heritability, cross-species differences, and behavior in the striatum, thalamus, and globus pallidus? I'm just going to walk you through the rationale to support the intuition behind this work. So say you have areas that are more heritable that are also positively correlated with cross-species differences. This means that more heritable areas in the human are also more expanded relative to, for example, the chimpanzee. So any areas that are positively associated with those ones can be inferred to be genetic and human specific, whereas negatively correlated areas can be associated with environmental and shared behaviors. On the other hand, if more heritable areas are actually more preserved across species, so they're negatively correlated with cross-species differences, then any associated behaviors can be inferred to be genetic and shared, whereas negatively correlated behaviors can be inferred to be environmental or human specific. And so the hypotheses underlying this work are that areas with a positive correlation between heritability and cross-species differences, meaning that more heritable areas are more expanded in the human, should be genetically mediated and associated with human-specific behaviors, whereas areas with a negative correlation between heritability and cross-species differences, meaning that more heritable areas are more preserved across species, should be associated with genetically mediated and shared behaviors. Another one, the caveat here is, of course, we are not zoologists, and we're only looking at heritability in the human, and we're only going to be looking at human behaviors as well. So it's difficult to obviously see how they translate to the non-human primates, but it's still interesting to try to make certain inferences about this. So the last point I want to address before getting into the methods and results is the quantification of subcortical anatomy. So, now that we understand why the striatum, thalamus, and globus pallidus are so relevant within a comparative neuroanatomy framework, we actually have to think about how to quantify properties in a sample size sufficiently large to be able to compute statistics. And so unlike the cortex, which is made of elegant and easily separable layers of cells, the cellular composition within subcortical structures is a little bit more chaotic. For instance, the striatum is named the corpus striatum because of the slender bridges of gray matter that extend through the internal capsule, giving it a striped appearance. And although 95% of it is made of medium spiny neurons, it also does have fast spiking GABAergic and cholinergic interneurons. The thalamus, on the other hand, is made of about 50 gray matter nuclei that are separated by myelinated fibers called lamella. And these nuclei all serve different functions and are fairly hard to identify in an MRI scan. So you can't really distinguish them from each other. And finally, the globus pallidus is made of large pallidal cells, but also small local circuit neurons and intermediate neurons. And because of the fact that the internal and external segment is separated by the medial medullary lamina, this structure has a very pale appearance in structural MRI scans. So in light of this, cellular and organizational complexity of these structures, surface-based measures can be very interesting because they can eventually be used as a proxy for cellular density or distribution in a particular structure, and morphometric measures are fairly easily extracted through computational tools in a large sample size. So the following work, therefore, examines vertex-wise surface area and displacement in the three structures of interest. So in order to get surface measures, the first step is to create a surface of the structure of interest. So this entails taking a mesh of a two-dimensional mesh of equidistant points that are connected by lines, and you deform it to fit your three-dimensional structure. You can then compute for each vertex on that surface, you can compute a surface area. So to obtain the surface area of one vertex, you add about 1/3 of the surface of each of the triangles connected to the vertex of interest. And then you can also obtain displacement. And so this is you obtain this by projecting the deformation vector at each vertex onto the unit normal vector of the surface normal at each vertex. So you do a dot product to determine the magnitude of the displacement in the direction perpendicular to the surface at each vertex. And just to further motivate why surface area and displacement should be examined separately, because normally you would assume that if the volume is larger, surface area and displacement will both expand, but they're still fairly uncorrelated, at least in the subcortex. So for the striatum, so these are histograms of the distributions, the vertex-wise correlations between surface area and displacement. And this in the striatum, these correlations average 0.04. In the thalamus, they average minus 0.02, and in the globus pallidus, they average minus 0.01. So it is quite relevant to examine these shape measurements separately. And so in order to examine this relationship between heritability, cross-species differences, and behavior in the structures of interest, we divided this into 3 different projects that were then related to each other by adapting the cortical surface-based spin test of Alexander Bloch from the cortex to the subcortex. So for the first part, to examine, the relationship between genetic versus environmental factors, we examined heritability and of the shape measures of these structures. And this project extended previous work done at the lab by Dr. Sejal Patel, examining shape measures of the hippocampus. And just as a reminder, heritability can be thought of as a quantification of nature versus nurture. And once again, more formally, it's the proportion of variance of a trait attributable to additive genetic as opposed to environmental effects. For us, in our case specifically, we were lucky we had access to an open source data set of twin and non-twin siblings. So we use a twin and non-twin sibling model to compute heritability, but there are other ways to look at heritability as well. And this statistical model in general is valuable because it's a prerequisite for genome-wide association studies. It's used to biologically validate new candidate endophenotypes. The bivariate model is a straightforward way to gauge the relationship between 2 traits. And in comparative anatomy, you can ask the question of, are heritable traits in humans expanded or preserved across species? And how does this relate to behavior? So we used 1,086 subjects, so 3 Tesla T1 weighted MRI scans from the Human Connectome projects. So they were healthy young adult twin and non-twin siblings. Images were pre-processed with MIG-B pipe and various steps of quality control, as well as removal of subjects without siblings. Removed our sample to 950 subjects, averaging about 29 years of age, with 517 to 433 female to male ratio. We then segmented the 3 structures of interest in all of our subjects using the maggot brain algorithm and quality control for structure-specific segmentation accuracy, reduced our sample size to 821, 865, and 950 subjects for the striatum, thalamus, and globus pallidus. And then, so once you have labeled these structures in each one of your subjects, you still need to create a mesh in each subject such that they all have the same amount of vertices across structures and a subject-specific surface area and displacement value associated with each vertex, because that's what allows you to compute statistics on your sample. So the maggot brain morpho setting allowed us to obtain surface area and displacement values for each subject in each structure on each vertex. And basically these values were residualized for sex and age before being input into a heritability model, which used the R package in OpenMX and extended previous scripts by Dr. Sejal Patel. And so once again, a heritability value that nears 100% means the variation of the trait is mostly controlled by genetic effects, whereas the heritability value nearing 0% means that the variation of the trait is mostly controlled by environmental effects. And so looking at the heritability of volumes, we found volume heritability. So for total brain volume, as well as for ipsilateral structure specific volumes, ranging from 81 to 94%, which confirms what we see in the literature. But given that we have all these shape measures and we want a more fine-grained information than just volumetric information, we looked at the heritability and vertex-wise surface area and displacement. So for surface area, these are the histograms of the distributions of structure-specific heritability values across vertices. So for instance, in the striatum, on average, vertex-wise surface area was 68% heritable, meaning that it was that the variation of these vertex-wise measurements, vertex-wise surface area, and the striatum was mostly controlled by genetic effects. This average was of 66% for the thalamus and 63% for the globus pallidus. Then looking at heritability of vertex-wise displacement, on average, these values were slightly lower with 62, 57, and 65% heritability averages for the striatum, thalamus, and globus pallidus. But mapping this back onto our surface measures, this is kind of what it looks like. And so basically in this graph in the top row, you have your vertex-wise surface area heritabilities. And then in the bottom row, you have the heritability values for vertex-wise displacement. On the left, you have your striatum, on the middle, the thalamus, and on the right, you have the globus pallidus. Values that are purple are less heritable, so they're nearing a heritability value of 40%, whereas values that are yellow are more heritable, so they're nearing a heritability of 100%. The views we are presenting for all of our structures are rostral, caudal, left lateral, right lateral, superior, and inferior. And then areas in white that you see, such as here, failed 5% false discovery rate correction. So I'm quickly going to walk you through surface area and displacement separately. But in the interest of time, I'm not going to list all the portions that are more or less heritable, but essentially, there are areas that are more heritable, areas that are less heritable. And then there is a cross structure bilateral symmetry that seems slightly disrupted in the inferior thalamus and lateral globus pallidus. For displacement, once again, there are more heritable areas and less heritable areas, and also bilateral symmetry in the heritability of vertex-wise displacement that's mostly disrupted in the superior caudate, lateral and ventral thalamus, and lateral globus pallidus. So to conclude this portion of this work, we found that the variance of subcortical vertex-wise shape measures is more strongly explained by additive genetic than environmental effects, with average heritability ranges of 57 to 68%. Shape measures are less heritable than volume measures, and there was a certain qualitative bilateral symmetry. So are there any questions for this portion of the project? All right, if not, I'll move on to the second project because I guess there's a lot of stuff to go through. So for the second project, we examined cross-species differences in these structures. And this portion was a lot of fun because it involved quite a lot of methodological innovation and creativity, thanks to Molar Chakravarty and Gabriel Deveni. And so basically what we did is that we took a human MRI template made by averaging the MRI scans of 5 human subjects. We took a chimpanzee MRI template made by Dr. Gabriel Deveni by averaging the MRI scans of 216 subjects. And so we ended up with 1 MRI template per species. We then manually labeled everything but the cortex in the cerebellum in order to obtain a little subcortical brain for each animal to make downstream computations less demanding. And then for the next step, Dr. Gabriel Deveni came up with a way to use ants registration in order to obtain a mapping from the human space to the chimpanzee space. So essentially what we did here is that we created an average brain between these two. So you can think of this as like this halfway space hybrid Frankenstein brain. And what was cool is that this allowed us to save the transformations going from the human to the average and from the chimpanzee to the average. But specifically, we were interested in surface measures, and we were in luck because we already had access to subcortical surface meshes on the human template that were created by Stephanie Tullo in 2018. And so all we had to do was apply the transformations saved from the ants registration pipelines onto the surfaces going from the human to the hybrid to the chimpanzee. And so now we went from having the striatum, thalamus, and globus pallidus surfaces on the human to having the same surfaces deformed to fit the chimpanzee, such that we have a one-to-one correspondence between vertices across species, which enabled cross-species comparisons. It is important to note that we only examined the most subtle nonlinear differences between structures in these 2 brains. So we adjusted for linear differences such as scaling, translations, and rotations in order to capture subtle shape differences and not volumetric differences. So in order to compare surface area measurements between the human and the chimpanzee, for each vertex, we took its value in the human and divided it by its surface area value in the chimpanzee. So we obtained a ratio of the relative expansion going from the chimpanzee to the human. And then for displacement, we just took the difference of the displacement on the human minus the displacement on the chimpanzee. So this essentially means that we computed the displacement going directly for each vertex going from the chimpanzee to the human. And so now, given all the allometry data that we've seen from the introduction, I quickly just want to show you what we found for the volumes, because it kind of confirmed what we already would suspect to happen in the subcortex. So in our data sets specifically, we found that total brain volume in our human template was 3.7 times larger than total brain volume in our chimpanzee template. And then for our ipsilateral subcortical structures, they were all volumetrically larger in the human relative to the chimpanzee, but this ratio was reduced. So this means that subcortical volumes don't expand as rapidly as total brain volumes, whereas the neocortex, as we've seen in the previous allometry studies, actually expands more rapidly than total brain volumes. So you could infer that the structures that we're examining in this work are more evolutionarily preserved, at least on a volumetric level. And so here we have our distributions of the areal and displacement differences that we found. The only thing I want you to pay attention to here is that for displacement, the values cover a more narrow range, because, as you can see from these histograms, and this is because we did have to adjust for residual affine effects here. And so this is what these graphs look like. Here we are plotting the color map as expansion and preservation, but it's a bidirectional one, just because it makes it a lot easier to see which areas are a lot larger in humans. So bear in mind that we did adjust for linear scaling differences, and that all these structures are still larger in the human. So areas in blue are, can be thought of as being more preserved across species, whereas areas in red can be thought of as being more expanded. And so zooming in on the surface area differences, we observed a pretty consistent bilateral symmetry qualitatively. And we see that the caudate is expanded, as well as rostral and caudal portions of the lateral thalamus and the rostral globus pallidus. On the other hand, we see that the putamen, lateral thalamus, and lateral globus pallidus seem more preserved across species. When looking at displacement differences, these values are more patchy. So, we're capturing some more subtle differences due to the residual affine correction. And we can see that these values are more pronounced in the left hemisphere. So to conclude this portion of the talk, we found that, in the surface area beyond the qualitative bilateral symmetry, we found an expanded caudate and preserved putamen, which is interesting because the caudate, although associated mostly with eye movement, it also does have prefrontal connections. So it takes input from the dorsolateral prefrontal cortex, which is, considered associated with higher order of cognition, whereas the putamen is usually associated with limb and trunk movement, so more lower order functions. And for the striatum specifically, the nucleus accumbens, we did not seem to follow any clear trend. And then we also found that the lateral thalamus and globus pallidus seemed more preserved across species. And finally, for our displacement differences, we found a higher magnitude in the left hemisphere. So are there any questions over here? Okay. If not,
I have a question.
Okay.
So when in your scaling differences, when you look at kind of the magnitude of the scale between going from human or chimp to human in any of these subcortical structures versus what you would expect in the cortex, can you give us a sense of kind of what that difference is and why you think those differences might be similar or different? So I mean, yeah.
For the volume.
Yeah, because it definitely the expansion certainly doesn't, at least in volume, and then the local expansion values are a bit under what you would see in the cortex, right? So
Exactly.
give a bit more interpretation to that and what that might mean functionally or in terms of adaptability.
Yeah, sure. So basically, total brain volume follows a specific speed of expanding. I mean, it just expands across species as mostly as they become more evolved, but also I guess as they become larger. But that doesn't necessarily mean that the volumes of all structures of the brain, expand at the exact same speed. So the cortex, as we've seen in the first allometry studies, the neocortex seems to actually expand faster relative to total brain volume. And so it's interesting because if the neocortex is expanding more than total brain volume, it means that other structures probably have to expand slower so that you arrive at your total brain volume kind of rate of change, right? And so this kind of makes sense that if the neocortex is expanding faster, you'd have some structures subcortically that would be expanding slower. And then you can kind of tie this into lower order and higher order functions because, like the neocortex is usually traditionally associated with functions that are, that make humans unique relative to other species, whereas the three structures, so the striatum, thalamus, and globus pallidus, have mostly, associated with motor functions that are kind of prevalent in primates as well. So that's what's interesting, is that this kind of does support, our volumetric results do support what we would expect to see in these structures. Yeah, does this answer?
Yeah, for sure. There's another question. Do you have any plans to quantitatively assess the bilateral symmetry of the changes you observe?
That would be really interesting. So the thing is, there isn't that much literature about lateralization in these structures as compared to the cortex. So I think I'd have to learn more about that, because I'm sure that there could be a way to adapt the spin test. We'd have to think of a way to adapt the spin test such that you actually, because right now the spin test, which I'll explain later, is performed bilaterally, right, for one structure. So you're going to examine the left and right striatum for one brain map compared to the left and right striatum for another brain map. So it'd be cool to adapt the Alexander Block spin test in order to actually compare two hemispheres to each other, which might have already been done. But that could be one quantitative way to compare the two, to see how correlated they are and whether that correlation is significant. Good question.
Yeah, can you do me a favor, Nadia? Can you just turn your camera off and on? You've been frozen on my Zoom for a bit.
Okay.
If that would help.
Yeah, I feel like my computer is on fire right now. Okay. Should I keep it off or should I try turning it back on again?
Try turning it back on. It's always nice to have you paired with the slides. Yeah, that's better. Thanks.
Okay, I'm moving. Okay, nice. So for the next portion of our project, we wanted to relate brain measures to behavior through a partially squares correlation analysis. And I'm sure some of you are already familiar with this technique, but I'll just briefly go over it anyways. And for the pre-processing steps, we did the exact same processing and pre-processing that we performed for the heritability study. The only difference was at this time, we only kept subjects that passed manual quality control for all three bilateral subjects. So this reduced our sample size to 801. So once again, for each subject, we have a bunch of vertices, and then for each vertex in each subject, we have our surface area and displacement measurement. And so the point of a partially squares correlation analysis is that it allows you to figure out which linear combinations of behavioral measures have the strongest impact on which linear combinations of brain variables. And so mathematically, you're performing a singular value decomposition of the covariance matrix between two sets of variables. So for our study, our brain matrix was a bunch of vertex-wise measures. So in the rows, you have your participants, and the columns you have each vertex. We looked at surface area and displacement separately here, just because they were examined separately for the previous two projects. And then through the Human Connectome project, we had access to an extensive battery of behavioral measures. So this included cognitive measures. We included one motor task and some, emotion related variables, as well as negative affect. And so basically we correlated, our brain measures with our behavior measures. You then perform a singular value decomposition. This was also all done in the PLS Python package for the PLS developed by Ross Martello. And then we obtained latent variables, each one made of a brain and of a behavior pattern. And so a permutation test was used to assess the significance of each latent variable, and values were bootstrapped and resampled in order to figure out which vertices contribute the most consistently to each latent variable. And so for the results, for the partially squares correlation analysis that we performed for vertex-wise surface area. We found 2 significant latent variables. For a second one, looking at vertex-wise displacement, we found 4 significant latent variables, and I'm only going to show you the results of the latent variables that explain the most covariance in each of these 2 runs, just so you have an idea of what the results of such an analysis look like. So over here, we have a plot where on the x-axis, you have the loadings of each behavior. We grade out all the behaviors for which the confidence intervals pass the 0 line. And then on the y-axis, we have the list of instruments that were used to measure certain behaviors. So instruments that are different shades of blue were associated with cognition. Instruments that are different red or warm colors were associated with emotion. And then the variables that appeared the most and loaded on the highest to this latent variable were associated with fluid intelligence and language. And so this was our areal latent variable that explained 64% of the covariance between vertex-wise surface area and our behaviors. So this is the bootstrap resampling ratio of the latent variable under examination. And it's just interesting to see, because this is how you map your latent variable back onto a surface map in order to be able to correlate it downstream with other surface maps, such as heritability maps or cross-species difference maps. And here it loaded very strongly and positively onto most of our brains, except for a few negative areas. And then for our latent variable that explained the most covariance and displacement, it explained 26% of the covariance, and it was associated the most with fluid intelligence, facial recognition, and language. So another higher order cognition related latent variable. And this is what the bootstrap resampling ratio for this one looked like. So just to summarize all the latent variables we obtained by the behaviors that explain them the most, we had 2 latent variables for surface area. I'm not going to, and the colors are associated with what category, so either cognition, emotion, or motor of the, what category the measurement fits into. And then for displacement, we had 4 latent variables. And this is a list that I'm not going to read out to you right now, but you can look at it later if you're interested. But yeah, so for the final, we have these 3 projects, and I started my talk going on about how we want to connect genetics versus environment, to evolution and adaptation, and to behavior. So the way that we actually related these 3 projects was by adapting the Alexander block cortical surface-based spin test to the sump cortex. And just to quickly explain for those less familiar with this test, how it works is that when you have 2 brain maps, you observe a specific correlation between them. And then what you do is that you take your brain map 1, project it onto a sphere, randomly permute it by a certain angle, and project it back onto the original brain map and look at how it correlates with your second brain map. And you do this a certain amount of times in order to artificially generate a distribution of your correlations. And then you look at where your observed correlation falls, and your p-value is essentially going to be associated with the area of all the randomly generated correspondence statistics that are higher than your observed correlation. It's important to know that this test is bilateral, that meaning that we examined it for this both left and right striatum were put together, left and right thalamus were examined together for this, and the left and right globus pallidus were examined for this. And we also had a lot of different pairwise correlations. So just as a reminder, we had 3 sets of brain maps. And for each brain map, we had vertex-wise surface area and displacement measures. So brain map 1, we had heritability measures for surface area displacement examined separately. Brain map 2, we looked at aerial and displacement differences separately between the human and the chimpanzee. And then for brain map 3, we had a total of 6 latent variables. So 2 latent variables for surface area. Each one has its own BSR, and then 4 latent variables for displacement. So each one of those has their own BSR. So in total, this is just a list of all the pairwise combinations of brain maps that we obtained and correlated with each other. And it resulted in a total of 5 correlations for vertex-wise surface area maps, 9 correlations for vertex-wise displacement maps, times 3 structures. So we had 42 correlations, which, I'm not going to walk you through every one, but basically false discovery rate correction across 42 comparisons with a 5% threshold left us with 32 significant correlations. And so just to quickly walk you through some of our direct correlations between cross-species differences and heritability, there weren't that many that were significant, but we did see some direct correlations between heritability and cross-species differences in the area of the striatum and the globus pallidus, and none in the thalamus. So these correlations were small but positive, which intuitively can imply that, you know, more heritable areas are more in these structures, in the area of these structures, are more expanded across species. And so any behaviors that are positively correlated with those areas can be inferred to be genetic and human specific, which is a relationship that we did capture between striatal area and areal latent variable 1 associated with fluid intelligence and language. So this latent variable was associated with higher order cognition. On the other hand, you know, negatively correlated behaviors could be, you know, inferred to be environmental or shared, which is a relationship that we captured between pallidal area and latent variable 2 associated with anger and delayed discounting. So with lower order cognition. And these findings are interesting given that the striatum is directly connected to the cortex, which is associated to higher order cognition, whereas the globus pallidus has no direct cortical connections. Expanding on these correlations, I'm going to walk you through some more correlations that we found. And just so you understand the downstream maps, all the bar plots in yellow represent correlations between heritability and a latent variable specific BSR, and all correlations in purple represent correlations between cross-species differences and latent variable specific BSR. And so you know, for surface area, we found a latent variable associated mostly with fluid intelligence and language. So this can be thought of as a higher order latent variable. And what was interesting is that it was correlated with heritable and expanded areas in the striatum, meaning that it was correlated with, you know, areas that are genetically controlled, but also very different in the human relative to the chimpanzee. And it was also correlated with less heritable and more preserved areas in the globus pallidus. So once again, we have this inverse relationship between, you know, between the correlations that might be related to connectivity to the cortex. Then we had another higher order latent variable that was associated with fluid intelligence and emotion recognition. This time it was in vertex-wise displacement. And what we found here was that it was positively correlated with areas that are expanded across all three structures. Although these correlations are below 0.1, they're still significant. And this is interesting because, you know, once again, higher order cognition, you know, is thought to be human specific. And then here it's correlated with areas that are more different in the human compared to the chimpanzee, that are expanded, sorry, in the human relative to the chimpanzee. And then when you look at more lower order latent variables, so we had one associated with anger and delayed discounting. And so this, you know, this in surface area, and this was interesting because it was correlated with heritable traits in the striatum, thalamus, and globus pallidus, but it was also correlated with more preserved areas in the thalamus and globus pallidus. So this is interesting because I feel like emoting, I guess you would have to speak to zoologists about how animals emote, but to me, it makes sense that this is associated with a more lower order function, and that, yes, it's more preserved across animals as well. And then we had another lower order cognitive, lower order latent variable. And this one was also correlated with heritable areas in the striatum, thalamus, and globus pallidus, and more preserved areas in the striatum and thalamus, and non-significant for the globus pallidus. So a similar trend to our previous one that also had, they're also both related to emotion. So that was an interesting thing to find. And so to summarize these results, we found that the variability in subcortical structure shape is more strongly explained by additive genetic than environmental effects. Shape measures were found to be less heritable than volume measures, which is also an interesting result. In our cross-species differences project, we found a preserved putamen and contracted caudate, which is interesting contracted, the slide is not contracted caudate, contracted putamen and expanded caudate, which makes sense given what we know about their functional roles in this their striatal roles. And then for our project relating these brain measures to behavior, we found 2 areal latent variables and 4 displacement latent variables. And our spin test relating all these 3 projects to each other allowed us to infer that cortical connectivity may influence correlation directionality, as we've seen in the differences between the striatum and the globus pallidus. We found higher order latent variables that are correlated with heritable and expanded areas, lower order latent variables that were associated with emotion and that were more heritable and preserved across species. And so future directions for this kind of work could examine plasticity. So for instance, where are the areas with high variance and low heritability? Or genetics, are the genes implicated in heritable areas that are also preserved across species? Are those genes phylogenetically preserved? That's a fairly, that for that you could definitely find the data to do that analysis. It would also be interesting to see how heritability, cross-species differences, and latent variable of behavior in the subcortical structure shape relate to subcortical cortical connectivity. And finally, it'd be interesting to, examine heritability and behavior in chimpanzees, because those 2 things were only examined in the human. So I'd like to warmly thank my lab and my wonderful supervisor, Malar Chakravarti, Gabriel Devani, Rehan Patel, and everybody else at the lab who's been unbelievably supportive, as well as all our external collaborators. Thank you very much.
Great talk, Nadia. There's a bit of time for questions if anyone has any. Maybe I will start off with one. So when you do the spin tests, and at the very end, the correlations are low, which is to be expected, right? How, do you have any thoughts on how? One thing, every time I see these results, I think like, oh, are we not getting an effective estimate of the relationship between maps? Because there might be some parts of the map that are well aligned and some parts that aren't, and the overwhelming majority might give us some of this correlation, but we're kind of missing some finer grain analysis about similarities and differences. And do you have any thoughts on that and how that can be achieved with the type of data that you have?
Well, I mean, so there's one student in our lab, Olivier Parent, who came up with a way that when you do the spin test, to also map on the residuals of these correlations to kind of try to see which areas contribute the most to the correlations. And we were discussing this with other students in the lab. But the thing is, areas for which the correlations are very low, they're going to have residual values that are super high for areas that have high heritability values. Or high cross-species difference values, for instance. So they're not necessarily, when a correlation is low, looking at the residuals does not necessarily reflect what parts of those brain maps contribute the most to that correlation, unfortunately. But it would be interesting. We do have some pretty high correlations as well that are up to 0.4 or minus 0.4. So it'd be interesting to examine the residuals for the correlations that have a larger magnitude, to kind of see what parts of these structures really contribute to these relationships.
It might even be worth doing it over a range of thresholds, right? Just to get a sense of how that's playing in, right?
Yeah,
that might be an interesting way to think about it. That's something that's interesting. Anyone else have any other thoughts or questions or anything like that? I don't see anything in the chat. Well, Nadia, I wanted to thank you for doing this talk. You did a great job. And thank you all for attending, and we'll see you all in the near future. Have a great day.